Can you see the screen?
Yes, ma'am.
Okay, fine. So, in today's class, we'll discuss about the topics that were covered in the last week, that is week 8, which is about isolation and purification of proteins, part 2. So in the last class we have studied about that how the different properties of proteins can be exploited for the separation of proteins, right? So in this class, just a minute, I think someone has joined. Hmm, yeah. So in this class we'll be continuing that part, and then we'll be seeing the rest of the different chromatographic techniques that were left. So, in the last class, we have studied about ion exchange chromatography, which exploits the property of charge of proteins, and hydrophobic interaction chromatography, which uses the property of the hydrophobicity of the proteins. So, we have studied that proteins are made up of hydrophobic amino acids as well as hydrophilic amino acids. The hydrophobic amino acids are present in the interior of the protein, and in the outside, the periphery, are the hydrophilic, that means water loving amino acids are there. So, with the help of some means, when we try to expose the hydrophobic amino acid residues, and that hydrophobic amino acid residues can be used for the separation of proteins. So, the property of charge and the property of hydrophobicity, these two properties we have studied. In today's class, we'll be seeing the property that is surface area and affinity. So, these two properties we'll be seeing. Just wait 1 minute. Yeah. So, first we'll just very briefly, very quickly discuss the important topics that were covered in the last class, in the last week, in the class 1. So, the first thing that we will be dealing today is gel filtration chromatography. Okay. So, um, basically, there are two kinds of proteins. Uh, First is globular proteins, and the other one is fibrous proteins. Okay. So, do you know what is the difference between globular proteins and fibrous protein? Anybody has any idea what are globular and what are fibrous proteins?
Ma'am, globular proteins are spherical in shape. They cover the least surface area, and fibers are like spread out proteins.
So, fibrous proteins are basically the elongated proteins, whereas globular proteins are spherical, like they are like globules, like sphere shaped proteins are there, and fibrous proteins are your elongated proteins, right? So, like these are your globular proteins, and extended form proteins, they are your fibrous proteins. Okay. Now, when we are talking about gel filtration chromatography, now filtration chromatography basically is used for the separation of globular proteins. So it works well when we want to separate globular proteins of different sizes, or we can also separate globular proteins from the fibrous proteins. So what actually happens is, okay, so Hari, you have raised your hand. What is the question you can ask? Okay, you wanted to see the answer. Okay, fine. As per the shape. Right. So, here we're talking about gel filtration chromatography. So, if this is a protein and when it gets properly folded, Then this is like a globular protein is formed. So this is a protein. So basically what happens in gel filtration chromatography is that proteins of different sizes, especially we are talking about here globular proteins, so globular proteins of different sizes can be separated from each other. How they can be separated? They can be separated because if you have a column that has beads of various sizes. So if I have a column, this is my column, okay, and this column is filled with a matrix in the form of beads. Okay, I have beads like this. These are my beads. And these are the beads in which there are pores, so that uh, the molecules, it can go inside it. So if this is my bead and there are like this pores in the beads, okay, so like this pores are there. This is just one bead that I am telling you. So like this there are many beads inside this column. So what happens is that your protein, which is mixed with the mobile phase, when it is passed through this column, then what happens? So molecules which are larger than the pore size of the bead, it will just flow through like this from the spaces, the voids, like this it will flow. But molecules which are of very small size, they will go like this inside the bead. Then molecules which are little bit bigger than this, then little more bigger molecules, and molecules which are even bigger, they will just flow through like this. So you can say that in gel filtration chromatography, which sized particles are eluted first? So if I say about the order of elution, okay, so what would be eluted first? Largest molecule or protein, whatever it is, largest size molecule would be eluted first, and last would be smallest sized molecule. Is it clear? The order of elution, that first the elution would be of the largest molecule, and the last your smallest size molecule would be eluted, because the molecules, it will go inside the pores which are of size lesser than the size of the pore, and the molecules which are, or proteins which are greater than the size of the pores, they will just pass through the void volume and it will just come in the flow through first. So this is what gel filtration chromatography. So basically in filtration, normal filtration, what happens that the particles which are smaller in size, they are eluted first, they get passed through the filter first. But here it is kind of a reverse filtration happens, because the molecules which are eluted first are the largest molecules, and then the smallest molecules comes in the last. So this is what gel filtration chromatography, very basic about gel filtration chromatography. So we'll see that what all was discussed in the class. So you can see that these are large pores, these are small pores. So what is happening here? You can see that the large size molecules, they are not able to pass through the pores, and hence they are eluted first, whereas the molecules which are of very small size, they get entrapped in the pores of the beads, and therefore they are eluted in the last, or later after the elution of the smaller larger size molecules. So now this is important. So again, as I was telling in the last class also, that this chromatography technique, the questions are very important for your competitive gate and dbt perspective. For dbt gate perspective, it is really very important. And there would be numericals from gel filtration chromatography, and the numericals would be mostly to determine the kd, or to determine the void volume, So these kinds of questions would be there. So important point to understand over here is this equation is important. Okay, this is important. This is very important. This is very important. And this is also important. This is very important. Okay, so determining the KD. And if you just remember even one equation, then you can just find out the values of any of them by just uh, exchanging them and rephrasing the equation. Anyways, so VT is the total volume. Okay, the total volume of the gel. VT is given by volume of the gel matrix, volume of the pores, and volume of the void. Void means the voids, the spaces. So from this we got to know that the KD is equals to VE minus VO by VI. Okay, this is what KD is. Now there are three important cases that takes place that we have to see. When KD is equals to 0, when KD is equals to 1, and when KD is equals to greater than, when KD is greater than 1. So our formula is KD is equals to VE minus VO by VI. Formula is KD is equals to VE minus VO by VI. So let us just do it here in our presentation. It would be easier. So our formula is KD is VE by VI. Elution volume, void volume, and pore. Now, when KD is equals to 0, if KD is equals to 0, then what will happen? This will go here. So, VE would be equals to V naught. Yes or no? When KD is equals to happen, VE will be equals to VO. That means the elution volume is equals to the void volume. Okay, so this is what void is. Okay, these are the void. This is what void is, the space. Fine. So when KD is equals to 0, you put 0 over here, it would be 0 VE minus VO by VI. So this means VE minus VO is equals to 0. That means VE is equals to VO. So when VE is equals to VO, that means the elution volume is equals to the void volume. That means the particle will not go inside the pore. It is completely outside the pore. And this would be eluted first. You can see here these analytes will be completely excluded from the column because it is not going inside the pore and they are completely excluded from the column. This is first case. Second case is when KD is equals to 1. Now you just put the value over here in this equation. This is our main equation. Okay. So it would be 1 is equals to VE minus VO by VI. This means VI is equals to VE minus VO. So this means elution volume would be VI plus VO. So for eluting, you need the void volume plus the pore also, the pore volume also. So these molecules, when whose KD is equals to 1, they will be completely inside the pore. These molecules, when KD is equals to 0, these analytes are completely excluded and they will be present completely inside the beads. Okay, so that is what they have said, that when KD is equals to 1, just a minute. Yeah. So when KD is equals to 1, this means elution volume is equals to void volume plus volume of the pores. That means these analytes will be completely in the pore of the column. Okay. And when KD is greater than 1, then these analytes would be adsorbed in the column matrix. So this is important. This is important. What happens at which KD value? This is one thing. Then in this class what we have studied, now this is important to see, If you understand this graph, then what, see, there are many, if you have gone through the chapter, so we have in the chapter they have discussed a lot many applications of gel filtration chromatography. So in all the applications, to understand it more scientifically, there is a graph, and in many places it is written that the graph shifts left side or right side or whatsoever. So you have to understand the graph and what the graph depicts, and then you can easily understand all the applications of chromatography, especially of this gel filtration chromatography, right? So this is the elution time. So the time, which is, if you have three molecules, one is the largest size, this is the smallest size, and this is a medium sized molecule, fine. So you see over here that the time which is taken by the largest molecule to come out of the column is smallest. Why? Because this molecule is not able to go inside the beads, because this molecule size is greater than that of the beads. Therefore, it is completely getting eluted. These are like, okay, some of them are going inside, some of them are coming also. This is very small, so it is going completely inside the beads. Therefore it will take the longest time to come out of the column. So this order of elution is very important, that to the leftmost largest analyte would be there, and to the rightmost smallest sized analyte would be there. Okay, important, leftmost largest size, rightmost smallest size, fine. Then these are the different kinds of matrix. So different kinds of matrix can be used. Cephadics is there, different varieties of matrix which have different range of pore sizes and bead sizes. And then basic operation of gel filtration chromatography, so how you have to pack the column, how you have to prepare the sample, then how the elution takes place. And this is very important, column regeneration. So what is the meaning of column regeneration is that you have a column that you have used for one round of gel filtration chromatography. Then you will not discard the column just after one use. You can regenerate the column and you can reuse it. Okay, so that is what they have mentioned, that after the analysis of analyte, gel filtration column is washed with the salt containing mobile phase, and then it is again washed with the medium. And this is important, how to store the column. So the column is stored at 4 degrees Celsius in the presence of 20% of alcohol containing sodium azide. So basically, column is stored at temperature of 4 degrees Celsius, and the chemical which is basically used for the storage of the columns is sodium azide. This is important, column regeneration. And how beautifully or how correctly you're doing the regeneration of the column will determine the efficiency of your next chromatographic experiment. Okay, so it is as important as performing the experiment to wash the column and properly store it so that it can be used for the next time, fine. Then then what we have discussed, then we have discussed the different applications. So applications part, we'll just take a little pause over here, and then we'll just see some of the questions, and then we'll come to the applications part. Okay, so just a minute. Okay, so so the first question is, these are very simple questions. Which chromatographic technique can be used to separate proteins of different diameters? Ion exchange, gel filtration, hydrophobic interaction, or partition chromatography? Tell the answer. It's easy.
Option B.
Option B, gel filtration chromatography. Yes. So, why gel filtration chromatography? Because here we are saying that proteins of different diameters, that means these are the proteins. First of all, what we can get, that they are of different diameters, that means they are round in shape. That is why diameter is coming. And then of different diameters means this has a different diameter, this has a different diameter, This has a different diameter. That means round. That means globular proteins of different sizes. So that can be separated with the help of gel filtration chromatography. The next
Excuse me,
question
ma'am.
is,
Ma'am, while doing this, if we don't know what all proteins are present in the mixture, say the target protein we extracted, there is also some impurity which also has the same size, then how we get to know that?
yeah, so actually, so when you are doing an experiment and when you are trying to separate the proteins, so this is not the first step that you do, okay? So basically, for this, I think the better explanation you will find in the last lecture, in the eighth lecture, in the very at the very last, sir has explained this also, that when you are performing the separation experimentally, then the first thing would be you try to do hydrophobic or ion exchange chromatography. So now what will happen, that when you are doing ion exchange chromatography, then you whatever proteins that you, now you know that what kind of exchanger you are using, cationic exchanger or anionic exchanger. So that is one tracking point, that you know that okay, so the proteins that are coming out, they are cations, or proteins that are inside, they are cations, depending on the type of exchanger you have used. So now you have one information about the proteins, that they are cationic or anionic, whatever is your this thing, research interest, but you don't know the type of the proteins. Then sequentially you can have an affinity chromatography. So now we'll discuss about the affinity chromatography also. So first you can have your ion exchange chromatography, then you can perform this gel filtration chromatography. So then you know that I want a protein, I don't know how many proteins are present in the mixture, but I want a protein which is known or which has some property to bind to a ligand X, suppose I know, but I don't know whether that, I don't know anything about the size or whatever it is. So in the end, when you are doing affinity chromatography, because you know that your column has that particular ligand X, and you want a protein which is against this X, so then you can just pass the column, and then you know which protein you have got. I'll explain you. Suppose we'll be explaining the affinity chromatography also. Suppose I want to produce an antibody. Okay, and I want this antibody against an antigen X. Now I don't know about the antibody, like whether it is really present in the mixture of proteins or not, but I know about this protein X, this antigen X, against which I want to isolate the antibody. Now I know that this protein uh, um, is negatively charged, let's say. So and I know that what kind of charged protein I want. Suppose I want to isolate positively charged proteins, because I know that my protein against which I want to raise the antibody is negatively charged. So I want the, so my protein of interest is positively charged. So with the help of since I want to isolate positively charged proteins, so I will be using which exchanger, anionic or cationic, cationic exchanger. Okay. um just a minute. So I will use a cationic exchanger. So what will happen, so now whatever is stuck in the in the column, that all is positively charged, but yet now we don't know that which amongst all the positively charged protein, which is our protein of interest. But now at least we have segregated the positively charged proteins from the negatively charged proteins. So now at least we have a pool of positively charged proteins. So first clue we got. Second clue, we know the size of this particular protein, this particular antigen. We know, let's say it is 300 kd, 300 kilodalton. So now we know that an antibody which is really effective against this 300 kd, so what should be the probable size of that antibody? It should not be like really less than 300 kd, otherwise it would not be very effective against it. So depending on that, we will design our gel filtration chromatography, because we don't know the size of our protein, but we know the size of the protein against which we want to isolate. So from here we perform gel filtration chromatography. So now again we have removed one level. Now we have the proteins which are of appropriate size. Now those appropriate size positively charged proteins, now we will have affinity chromatography where we have a matrix, and that matrix has X. And now when we will pass the third filter, and then whatever is interacting with the X which is present on the matrix, that can be the protein of interest. Is it clear? Is it clear, or
Yes,
you
ma'am.
have any confusion you can ask?
Yes, ma'am. Clear.
Hmm. So the basic concept is that, so here we are individually studying each and every technique, right? We are studying about hydrophobic interaction, we are studying about ionic exchange chromatography. But when you are doing an experiment and when you want to isolate and purify a protein, you don't just use one technique. You use number of techniques depending upon your experiment and your like what you want to isolate and what you want to purify. So when you are doing separation, so separation does not involve one step. Separation is a multi-step process. There are numerous steps which are involved in separation and purification of proteins. Okay, so now, have we discussed this question? No, just a minute. Okay, so in gel filtration chromatography, so this is filling the blanks. Dash molecules are excluded while dash molecules are retained in the pores of the matrix. Which molecules are excluded and which molecules gets into the pores of the matrix? Which molecules are excluded? Tell.
Ma'am, large molecules are excluded and small ones are retained.
Hmm. So the answer would be B. Large molecules are excluded while smaller sized molecules are retained in the pores of the matrix. Okay. So next question is, while performing gel filtration chromatography, Sunita used a bead of 25 nanometers. Okay. What does this imply? So the bead size that she has used is the bead. This is the bead. This is 25 nanometers. Okay. So A is smaller molecules will pass through the beads. Molecules above 25 nanometer will be eluted and molecules below 25 nanometer will be retained. Molecules less than 25 nanometer would be eluted first, or molecules which are larger than 25 nanometer will stick in the bead, and molecules lesser than 25 nanometer will easily pass. So tell me the answer, but although there are very few people today, but I don't want Karthik to tell the answer. The rest three you may try, rest four, or like how many are there? Can you try? You want to try this question? We have already discussed and I think we have solved the question also. Anybody? Nobody knows the answer? Just choose one option. Try try to answer the question. So if the bead is 25 nanometers, so then what kind of molecules would be eluted and what kind of molecules will be retained in the bead? Think, try to think at least. No? B. Okay, so Swati has said B. Molecules above will be. Yes, very good. Swati, you gave absolutely correct answer. So what we are saying, that if this bead is 25 nanometers, then molecules which are above 25 nanometers would be eluted obviously, because if this is a molecule and this is the pore size, this cannot go inside the pore. It's a basic logic. So obviously it would be eluted. But if there is another molecule which is suppose of this size, obviously it can go inside the bead because there are pores. So Molecules which are above 25 nanometer, they will be eluted, and molecules which are below 25 nanometer will be retained. Actually it should be retained inside the bead. Fine. Next question is, and this is open to all. During gel filtration chromatography, largest elution volume is observed for. Largest elution volume is for largest molecule, smallest molecule. Elution volume does not depends on the size, or for Kd is equals to one molecule. Anybody can answer the question.
Option A.
During gel filtration, largest elution volume. Yes. So largest elution volume is no. Uh, it should be B. Now, why it would be B? Okay, just 1 minute. Where is the formula? Elution volume. Kd is equals to VE. See, this is the elution volume formula. Elution volume is equals to the void volume. So elution volume is related to the void volume plus the volume of the pores. Okay. So when we have largest molecules, so largest molecules, the VI would be zero only. Why? Because they are not going inside the pore. So their VE is only equals to the void volume. Whereas molecules which are smallest in size, so their elution volume would be more, because then there is VI factor also. Is it clear? Is it clear or it is not clear?
Not clear, ma'am.
So you have to understand that. Let me just write the formula. V is equals to V naught VE is equals to V naught plus KD VI. So this is the formula of elution volume. So elution volume depends upon the void volume and the volume of the pores. For molecules which are like, for the largest molecules, these are not going inside the pore. Okay, they are not going inside the pore. And because they are not going inside the pore, there is no this factor. So this is their void volume, their elution volume only depends on the void volume. But smallest particles, they are going inside the pore, so their elution volume would be more. And also because smallest particles are traveling more than the larger particles, so obviously they will have more elution volume, they will carry more volume. So the answer of this question is that gel filtration chromatography, largest solution volume is observed for smallest molecules. Then we come to um, lecture 2. So in lecture 2, basically there was a video demonstration that just to show you and to explain you uh, um, how gel filtration chromatography it takes place in experimentally, and then comes the applications of gel filtration chromatography. Now you have to understand the graph that we have discussed. So if this is the elution time, this is the elution time, and this was absorbance. So the first peak that we are getting, three peaks. So this peak is for the largest molecule, then medium sized molecule, and this is smallest molecule. Now this is important. So this, why I am saying again and again, because when we'll be dealing the applications part, then this graph and the properties of the graph will only help to understand it, right? So we'll just quickly go through the applications and then we'll move to the next, and then we'll see the questions. Okay, so now we know that what is, so if you have any doubt regarding the basic concept of gel filtration chromatography, you can ask me. Okay, So the main thing to understand and to know about gel filtration. So gel filtration chromatography is also known as size exclusion chromatography. So you might get questions where they have not used gel filtration chromatography term at all, and they have used size exclusion chromatography. So you need to know ki both the things are same only. It's just that gel filtration chromatography has a lot many names. So the main thing is that globular proteins, which are spherical in nature, obviously because they're globular, are of different sizes. And this property of different size is exploited to separate molecules based on their size. That is what gel filtration chromatography is there. So I don't know whether you have seen so there are certain balls, when you put it in water, they kind of swell up. They take the water and they kind of swell up. They are transparent like water balls that we really use in for decoration purposes. During Diwali, we put diya and we put water and we put those balls. Do we have those? Do you have any idea? Can you recall what I'm talking about?
Yes, ma'am.
So transparent balls, like they are very small, very tiny balls are there, but as soon as you put them in water, they kind of swell up and they become of good size. So just like those balls which are of very small size, they come in small packet, and when you put them in water, they kind of swell up. In a very similar way, the beads are also there. So basically, you put bead inside the matrix and then they swell up, and you can just try to correlate with those balls, those transparent balls you have. So just like that, those beads also kind of swell up inside the matrix, and then they completely fill the matrix. Something like that, you can just relate it. Anyways, so the first application of gel filtration chromatography is the determination of molecular weight. So molecular weight, how molecular weight can be determined? Because molecular weight is directly proportional to the radius of the molecule. So okay, so uh, you can have just a minute. Okay. So here you can see that the molecular weight and size of a protein is related to the shape of the molecule. The relationship is that molecular weight is directly proportional to the radius. Okay. So radius of gyration means that we are taking radius from the center of that particular molecule. So that means, because these whatever molecules, whatever analytes we have, they are not they cannot be completely circle also, because they are spherical in shape, they are globular in shape, but they're not exactly completely circular also. So that's why we just not say radius, we say radius of gyration. That means we are taking the radius from the center of the mass of that particular object, from the center of the object, which is a globular shaped object. That is why we have used gyration and not just the radius. Okay, we take the term as radius of gyration. So basically, we can determine the molecular weight with the help of gel filtration chromatography. Then we can use the oligomeric status. That means whether the particular protein, it is present in a monomeric form, or it is present in a dimeric form or tetrameric form. So that also you can determine with the help of gel filtration chromatography. This is the formula. If you have any questions, you can solve with the help of this formula also. So basically, you have to understand that suppose uh, you so you do you have the basic idea of how SDS works? So SDS basically is an anionic detergent. So the full form is sodium dodecyl sulfate. So it imparts negative charge to the proteins. And there are, when you are doing electrophoresis for proteins, there are two kinds of uh, electrophoresis that you can do. Sorry. The first one is a native page, and second one is an SDS. So in native page, what happens is that the conformation of the proteins is preserved. That means your protein is not denatured. When we are talking about native page, okay, we are talking about native page. In native page, there is no denaturation of proteins. Whereas when we talk about SDS page, so here proteins are denatured. Okay. So this denaturing of proteins occurs because of the detergent that we have used, that is sodium dodecyl sulfate SDS, which is a negatively charged anionic detergent, and it imparts negative charge to the proteins. So if a protein is of 50 kilodaltons, and with the help of SDS page you got a band at 10 kilodalton, then can you tell the oligomeric status of this protein? Can you tell? Anybody? The total weight of protein is, mass of protein is 50 kilodalton. And with the help of electrophoresis, you did your electrophoresis. So basically, for DNA, you do agarose gel electrophoresis, and for the proteins we do polyacrylate gel electrophoresis page. This page can be a native page or an SDS page. So by after doing your page, you did your you run the gel, and then you saw that the band which is coming, it is coming at 10 kilodalton. So what you can say, what can be concluded out of it? Tell me what can be concluded out of it.
Ma'am, the protein is made of five subunits, I think.
Yes. So this is total, the total weight is 50 kilodalton. The total mass is 50 kilodalton, and you're getting a band of 10. That means 110, another, then another 1, 2, 3, 4, and 5. That means there are 5 subunits, each of 10 kilodaltons, which are present. So it's like a pentameric protein, pentamer. So like this you can also have uh, one band at 10, one band at 20, one band at 30. Okay, one band at 20 and one band at 30. So it depends, like the questions can be of different kinds, but what I'm trying to say is that the oligomeric status of the protein uh, can also be determined with the help of gel filtration for chromatography, because once we determine the weight, what is the weight, molecular weight, then with the help of SDS page and native page we can determine the oligomeric status. This is important, this formula is important. Then another application is to study the protein folding. Obviously, how you can study the protein folding? Because um as the protein is getting unfolded, what is happening? As
Excuse
the
me, ma'am.
protein is getting unfolded, it is losing its globular structure. You can see unfolded protein has the smallest elution volume, and it will be eluted at first. Why? Because this is a native protein. Okay, it is very completely globular and very compact structure. You are providing urea, so for denaturation urea is provided, urea is given. So you're giving urea, and this urea is causing the unfolding of the protein. Again, you're giving more urea, more unfolding occurs. So now, this, because of the unfolding uh, process which is taking place, this protein is now coming to an extended conformation. From the globular conformation, it is coming to an extended conformation. And because it is becoming an extended conformation, that is why its size is increasing. Now it can no longer go inside the beads, and therefore it will be eluted first. So the unfolded protein will have the graph at the leftmost corner, and the native protein will have the graph at rightmost corner. So this is how we can study the protein folding. Then,
Excuse me, ma'am.
yeah.
Ma'am, I had a doubt regarding native page.
Okay.
Ma'am, in SDS page, we use SDS to give the protein negative charge. So it will only move towards the positive electrode. But in native page, we are not giving any charge to the protein. So if the protein is, say, positively charged, it will start moving towards the opposite end, right? So how do we determine, uh, I mean, what should we do in that case?
So, one thing that you have to understand is that the conformation, the folding of the amino acids, those are the factors that separates the proteins. Okay. Now, basic thing to understand over here is that the gel that we are using for the SDS page and for the native page, that is not, that is denatured in the case of SDS, and that is not denatured in the case of native page. So here it depends. See, you have to have a little bit of information about the proteins, or uh, the protein can have different charges. It can either be a positively charged protein, or it can be a negatively charged protein, or it can be a neutral protein. Now, whatever movement that we are getting, so that actually kind of helps us to determine about, to get the information of the protein, first thing. And second thing is to determine the size of the protein. Second thing which is very important is that it is not only the charge, that is the positive and the negative charge, that determines the run of the protein, but the overtaking phenomena, the overtaking concept, or the concept which is more overpowering, is the size of the proteins. So when you have a native conformation, so in native conformation what happens, that their exact quaternary structure is preserved. So because of the preserve of that quaternary structure, the size, the molecular weight of the different proteins will overpower the contribution that happens towards the charge. So there has to be a balance. So there's actually a formula. I don't remember the formula for determination of the size. I have to check. If you can just remind me in the next class, I'll come up with the formula also. So I think that formula will be able to explain you in a better way that how there is a balance that takes place between the charge and the molecular size of the proteins, that helps in the this thing, determination of the molecular weight. Because when we are doing SDS, then the separation is only occurring because of the difference in their molecular weights, because all the proteins are negatively charged only. But here, because there's no negative charge that we have imparted artificially to the proteins. The proteins have their inherent charge, whether it can be a positive or it can be a negative. So here the contributing factors are charge as well as the molecular size. So the balance between the two determines the size of the protein, and that is how the gel is run. So I don't remember the formula exactly. I have to see and check. If you can give me 1 minute, I can just try to see. Otherwise, we can discuss the formula in the next class also. I think then it would be easier for you to understand the principle and how it works. Okay. So you just remind me in the next class. I will come up with that formula, because that formula will only explain. Do you have? Y- do you guys have Pathfinder right now with you? Anybody?
Yes, ma'am. I was taking out that only.
Because I think there is a the formula is given.
Yes, ma'am.
I don't have it right now. If you have, you can share it to us. Otherwise, we'll discuss it in the next class, or maybe afterwards, if we have some time left after completing everything, then we can discuss that. Okay. I'll just just check the formula. I just it is there in Pathfinder, but I am not able to recall the formula. Whatever it is. Okay. So next is protein ligand binding. So again, the same principle, that when there is only ligand is present or only protein is there, then obviously the size is small, so the graph would be at the right side. But when the ligand and protein both are there, then obviously the size is increasing, and because the size is increasing, so the graph is shifting towards the left side. So these are some of the applications. There are many applications actually, but let us just quickly move to the questions, because we are still in the second chapter, and there are so many chapters in the last class. So, okay, question number 5. If we have two different proteins, keratin and hemoglobin, Amongst the two, which will be eluted first? Give reasons as well. So, reason you have to give. Reason I have not put up in the MCQ. So you have two proteins, keratin and hemoglobin. So which would be eluted first? What do you think? Which protein would be eluted? You need to have the properties of keratin and hemoglobin.
Keratin will be eluted first because it's a fibrous protein and it won't stick inside the beads.
Correct. So basically, here the question is very easy. Just in spite of giving the technical name, I have given an example of that. So keratin is actually a fibrous protein, and hemoglobin is a globular protein. Okay. So obviously, this gel filtration chromatography works well for globular proteins. So globular proteins will be excuse me, eluted last, because they can go inside the beads. And see, it depends on the size of the pores that you have used. So there can be a chance that even the globular proteins cannot go inside the bead, because the bead size is way too small than the size of the hemoglobin. But there can be a possibility that it can go also, but for fibrous protein there is no such possibility. So obviously, amongst the two, the more chances are that keratin would be eluted the first. Next question is, for studying the protein folding using gel filtration, which is the most commonly reagent used? Which is the reagent which is used for unfolding the protein? The most common reagent using gel filtration chromatography?
Urea.
Urea. So urea is what we generally use. The different concentration of urea will determine the different levels of protein folding, protein unfolding. So this is urea. Next question is, the extended proteins in gel filtration are observed inside the pores of the beads. They're adsorbed on the bead surface. Some are inside and some are outside. Some are, and none of the above. What is the answer? Extended proteins.
None of the above, ma'am.
Yes, the answer is none of the above. Why? Because extended proteins are neither adsorbed nor they are present inside the beads, nor there is a possibility that some can be inside, some can be outside. Extended proteins will be completely out of the pore, and if this is a column, if this is the column and these are the beads, so extended proteins would be completely eluted out from the column. They cannot go inside, they cannot be like some are inside, some are outside. So the answer is none of the above. Fine. So now we come to the affinity chromatography. I think 1 hour is already complete. Yeah. So this 1 hour we have to give for affinity. There are at least um, there are four chapters, I guess, here. Yeah. So now we come to affinity chromatography. But before affinity chromatography, I think there was antibody production, which is more important. So basically, affinity chromatography is, I think by now you must have a little idea of what is affinity chromatography. So, affinity chromatography means that whatever protein that you want to separate, you want to isolate, that protein has a special interaction interacting partner, or that protein is known to bind to a specific ligand. Suppose this is a protein X and we know that this protein binds to, say, NTA column, because this protein has nickel in it. So this interaction property, or because we know that this protein will bind to this column, so this property is exploited to isolate this protein, let's say A. So that is why the name is affinity chromatography, because it depends on the affinity of a protein to bind to the matrix of the column, affinity of the protein to bind to the column. So what is there in this chromatography is that this is your column and this is your matrix. Fine, this is your matrix. And this matrix is special because it has uh, receptors for a particular ligand, this is the receptors which are present. Okay. Now, when you give a mixture of proteins like this, these are the different kinds of particles which are present, and this. So, this ligand can bind to the receptor, and rest all other, this, this, other, they will be eluted out. So, the specific binding of the ligand to the receptor, or not just ligand and receptor is just an example to tell you, to make you understand the concept, but the specific affinity of a particular protein to the matrix will be exploited for affinity chromatography. Okay. Now, we'll see what affinity chromatography is, what we have learned in the last week. So, you can see affinity chromatography works on the principle of mutual recognition force between ligand and receptor. So basically, the major determinant is shape complementarity. It's very important. Why? Because see, this is your protein and this is the receptor. If there would be shape complementarity, then only other forces can be able to bind with it. So the main responsible force is shape complementarity, the primary force, and others are electrostatic, hydrogen bonding, van der Waals forces. Other kinds of forces are present. So basically, here the Kd value, again you have to understand about the Kd value, and then what we have discussed in this then there are various advantages. This is all like, just you have to just read it, that because this is very specific, because so basically this affinity chromatography, when you are doing an experiment, so you try to do affinity chromatography at a very later stage, or probably at the last stage also, because it has a lot of specificity. So after all the filters, when you have some chunks of protein mixture left, and then you know that you want to isolate a protein which is specific to a receptor Y, then you can easily perform affinity chromatography, and it will give you a lot of specificity. Then it gives a high level of purification, it is very reproducible, and it is easy to perform. These are the basic advantages of affinity chromatography. Now there can be different types of affinity chromatography. The first one is the bioaffinity chromatography, and the other is the pseudo affinity chromatography. So bioaffinity means that biological known interactions that occur in nature, that interactions are utilized or exploited for the creation of affinity columns. And then you have pseudo affinity, means they have affinity to a dye, affinity to a metal. So like there's different kinds of affinity chromatography is there. So I think basic we have studied, so we'll just try to do some questions, and then we'll get back to the even deeper into it. Okay, so the first question is, which chromatography works on the, very easy it is though, which chromatography works on the interaction between receptor and its ligand? Affinity, gel filtration, ion exchange, HIC. This is okay, A. Yes, affinity chromatography. Actually, because I'm teaching, so it's difficult for me to continuously switch and see what, whether you have given the answers in the messages or not. So if it is possible, you can just say the answer. It would be a little more convenient for me, because then I have to constantly switch between the two screens. Anyway, so Swati has given the right answer, that affinity chromatography works on the interaction between receptor and its ligand. The next question is, dash is the primary recognition force between the receptor and ligand in affinity chromatography? Hydrogen binding, electrostatic, van der Waals forces, shape complementarity. What is the answer? What is the answer of the ninth question? Tell. Dash is the primary recognition force between the receptor and ligand in affinity chromatography. What is the primary recognition force?
Ma'am, electrostatic interaction.
No. The primary recognition force between the receptor and the ligand is shape complementarity. Just we have discussed. So, if this is your receptor, this is a diagram from the PDF itself, and this is your, let's say, ligand. So it doesn't matter if it is, it might have electrostatic interaction between them, or there might be the possibility of hydrogen bonding, but still if it goes inside, this interaction would not be very proper, because it does not fits well in the shape, right? So the first and the primary force that kind of recognizes and that kind of makes it better is the complementary shape to it. I mean, it's not a very good diagram, but just wanted to tell you that if so many different forms of molecules, whether it is this or any other shape, it might have uh, it might exhibit electrostatic interaction or hydrogen bonding and all of that, but because the shape is so different, so it will not have a proper binding or proper packaging. So that's why the first thing is, then it should have a shape complementarity with the receptor, and then of course whatever other factors that helps in the binding, it will also contribute. The first thing is it should have a complementary shape. Fine. Next question is, in bioaffinity chromatography, what kind of interactions are exploited? Antigen antibody interactions, enzyme substrate that forms a product, enzyme substrate that does not form the product, A and B and A and C. Try to answer the question.
Ma'am, both A and B.
No, this is a wrong answer. The correct answer is both A and C. Now think why it's not B but it's C. Why you think it should be B? It should be C and not B in this question.
Ma'am, I thought from the product we can determine that the protein is present there, like ELISA or something. That's why I said.
No, that is, see, that is a different thing altogether. Okay, here, see, the thing is, you want to, that protein, whatever protein is there, that is binding to the enzyme, that protein can have n number of consequences and it can form a product, but it is not the product that you want to isolate, it is the protein that you want to isolate, because you have to, whatever, depending upon your experiment, you need the protein, you do not need the product, because you might not need the protein, take the protein for the production of that product itself. You might want this protein for some other purposes, because if you are taking out the protein for the product formation, then what is novel in here? So if you have a column and whatever enzyme substrate reactions or whatever is there, suppose substrate is there, or enzyme is there, whatever one of them is there, and the other one, when it combines with it and it forms another product, then what is happening? Your protein of interest is gone, because you wanted to isolate this, but what you are getting is this products. You don't want this, you want the protein itself. So that is why it is very important to understand that when we are doing bioaffinity chromatography, so bioaffinity means that affinity chromatography that is using the biological interactions, the biological interactions that exist in the nature. So those interactions are used up and then affinity chromatography is performed. So antigen antibody interaction is one kind of bio interaction. Another kind of interaction is enzyme substrate reaction. But the enzyme substrate reaction should be in a way that it is not able to form the product. Because if it is forming the product, then you are actually separating the product and not the protein. Is it clear?
Yes, ma'am.
Hmm. Then next question is, which type of affinity chromatography has non-breakable interaction with the between the matrix and the ligand? Bioaffinity, covalent, metal affinity, diaffinity chromatography. Think and answer.
Ma'am, covalent chromatography.
Yeah, covalent chromatography. So, if you have really gone through the chapter, so basically almost all of the affinity chromatography, the interactions, so almost all of the interactions are irreversible interactions. So reversible interaction means that they can be broken up, they can be changed to its native form, the original form again. But there is another kind of affinity chromatography where the interaction is non-breakable. It is irreversible interactions that takes place. So amongst the options, irreversible kind of bond is formed only in the case of covalent bonds. So covalent bonds, covalent bonds, so they are irreversible breaking. They are not break. They cannot be broken up. So here the answer would be B. That is covalent chromatography, because bonds are not breakable. So the kind of chromatography where uh, between the matrix and the protein there is covalent interaction that takes place, that is your covalent chromatography. And covalent chromatography is a non-breakable kind of an interaction. So then we come to the next lecture, lecture 4. So this lecture 4 is actually a little bit important. Okay, now, so far we have learned about size exclusion chromatography. We have learned about affinity chromatography. We are still reading about affinity chromatography, and like I always told, I told in this class also that these are very important topics per se, your exam point of view, for your competitive exams point of view, whether you're giving gate or you're giving net or you're giving DBT, whatever it is. Another very important topic is antibody production. Now, antibody production is not only important for your exams point of view, but if any of you who want to pursue higher education, who wants to go for research, which you should go, so then also antibody production is very important, because in a lot of immunology labs, you have to generate antibodies by yourselves. So a lot of times I don't do, but other people, they do generate antibodies, and then they use the generated antibodies. They don't really purchase the antibodies. Not every time that you purchase the antibodies. So basically, when you're talking about affinity chromatography, so we already discussed that the interaction can be of antigen antibody interaction. Okay, so suppose this is the antibody or the antigen, whatever one of them is, which has to bound to the matrix. So that is your receptor. So that means the receptor for the matrix, how to create the or to generate the receptor of the matrix, that is what is explained in this part, generation of the receptor. So the receptor can be your antibody also. Suppose if this is your column, this is your column, and this is your column, and these are your antibody. Okay, antibody, antibody, antibody. And antigen is what? what is the what is an antigen? As in, it's a protein or a carbohydrate or a nucleic acid. What is an antigen? Tell me.
Ma'am,
Antigen is
usually protein, but it can be combination of sugar protein, lipid protein.
antigen is a protein. So if you have a mixture of proteins which has your antigen also, which you want to isolate, so when you give it, so that antigen will bind to it, and that is how, with the help of this antigen antibody interaction, you are able to isolate the antigen of your interest. Now, to suppose you not every antibody, not every antibody can be available, or even if it is available, you can generate it by yourself also. So generation of receptor means that, if for example we have antigen antibody reaction, so how to generate this antibody, that we will now see. Okay, so basically what you have to do is that you have to generate the antibody. Now, antibody is generated in response to a particular antigen, against a particular antigen. So, basically, to generate antibody, first you have to prepare the antigen. You can prepare the antigen with the help of this genetic engineering or whatever purposes. So, you have prepared an antigen, and then that antigen basically can be given to an animal. Now, one thing important over here is, see, this generation of antibody. First thing is that you have to prepare the antigen, then you can prepare the antigen, then how to isolate it, then how to purify it. So, this is all. Basically, the thing is that you have to prepare the antigen. So, now when you have prepared the antigen, then the next thing is the preparation of immunogen. Now, can you tell me what is the difference between an antigen and an immunogen?
Ma'am, immunogen produces an immune response, but an antigen may or may not produce an immune response. If it produces an immune response, then it's an immunogen.
Yes. So basically, antigen is anything, just a thing that can bind to the antibody. Okay, that is just antigen. Antigen is a protein which binds to the antibody. It is not necessary that it will always produce an immune response. So sometimes, see, the immunogenicity of any antigen depends on a lot of factors. So it depends on the size of the protein, it depends on the molecular weight, it depends on the complexity of the protein. So there are many factors that contributes to the immunogenicity of a particular antigen. So if an antigen by itself is not that immunogen, that means if an antigen by itself is not very much capable of producing a very good response, then what we can do is that we can tag the antigen with certain other molecule, so that now that fusion will have a very good will produce a very good immune response. Okay, so that particular thing which is added to the antigen, which will enhance the immune response of that particular antigen, that is known as adjuvant. Is it clear what is an adjuvant? So basically, if this is your antigen, this is your antigen. Okay, and this antigen by itself is not very efficient in the manner that it cannot create a very well elaborated immune response by itself, because of several reasons. It might be a very small, it might be very less complex, whatever it is. But we want this particular antigen to create a very good immune response. So what we can do, we can tag this particular antigen with some other molecule, and this molecule will enhance the antigenicity of this particular antigen. So this is known as adjuvant. Okay.
Ma'am, then what about hapten?
Haptens, see, haptens and adjuvants are different. Why? Because you will never interact your antigen, you will never tag your antigen with haptens. You will only tag your antigen with adjuvants. Why? Why you think you will not tag your antigen with haptens? What do you think?
Ma'am, the hapten may also act like a antigen itself.
Yeah. So basically you have to understand. So basically hapten, when you bind hapten with any particular antibody or a particular protein, so it will enhance its response, but it will not have a lot of antigenicity of its own. Okay, so this hapten basically it combines with antibodies, and it will enhance the immune response, but it will not have any antigenicity of its own. So it will not help the antigen to exaggerate the response. Basically, it's a kind of a carrier molecule. Hapten is nothing but it is a kind of carrier molecule only. So for combining your antigen with a particular agent, a particular molecule, that is contributing in enhancing the antigenicity of the antigen. That is the function of adjuvant, and that adjuvant can only do. Hapten is just a carrier molecule. Okay, so we were discussing about the generation of antibodies. So now we first prepared the antigen, and then we make it more antigenic by making it an immunogen, and then we will give this antigen to a particular organism, maybe a rabbit or a mouse. We give it to rabbit or mouse. After giving, so now because we have given antigen, so then what will happen? Because every body has an immune response, so that immune response will produce antibodies, and then we will collect those antibodies, matlab we will collect the blood and we will prepare the serum, and then we will isolate the antibodies from it. Now the process that they have given over here is basically for the production of polyclonal antibodies, which are specific to a number of antigens. It is not for monoclonal antibodies. For monoclonal antibodies, the procedure is different. Have you studied about monoclonal antibodies and how to generate monoclonal antibodies?
Yes, ma'am.
So, which media is used there? Selective media. Three components that are needed. Does anybody remember? Anyways, you can read about it. So, there are three things. So, the media is known as HAT: hypoxanthine, aminopectin, and thymidine. These are the three things which are mainly, which are very important when we are producing monoclonal antibodies. Anyways, that is beyond the scope of the chapter. So now we have produced the antibodies. So for production of antibodies, first thing what we have to do, we have to prepare the antigen. If the antigen is available, well enough. If the antigen is not available, then you have to produce the antigen with the help of RDT technology. Then to improve its antigenicity, you have to combine it to a molecule such as adjuvant, and so that its immunogenicity is increased. And after that you will give that particular antigen to then to either mouse or rat, or depending upon against which you want to raise. And then you will give some booster doses also, and then you will isolate the blood, and then you will prepare the you can collect the antibodies. So this is how the production of antibodies takes place. And another rest, some other things also, you can just go through them. These are also important, but you can just go through them. We don't really have that much time that we can discuss each and every point in very much detail, but this was just a gist that how basically antibody production, polyclonal antibody production takes place. But in general, if you talk about competitive exam point of view, then monoclonal antibody production is more important than studying the polyclonal antibody production. It is also important, but more questions are covered from the monoclonal antibody part. So you should have a very thorough knowledge about that portion. That is very important. Actually, some questions from chromatography or monoclonal antibody, it always comes in the examinations. So if you have read about monoclonal antibody, you can read. And if you have any problem regarding monoclonal antibody production, we can ask me also. We can discuss in subsequent lectures, in the next lecture maybe. So now we'll do some questions again. Okay. Identify the correct, this just we have studied. Identify the correct statement in regard to the generation of an immunogen. So, for the generation of immunogen, what we do? Antigen is mixed with adjuvant, antigen is mixed with another antigen to make it polyclonal, antigen is injected into the animal, or none of the above. So, how immunogens are produced?
Ma'am, A.
A, yeah, antigen is mixed with adjuvant.
Ma'am,
Okay.
I had a doubt. Ma'am, the adjuvant can only increase the immunogenicity, right? It can't make uh, a antigen which cannot generate an immune response into an immunogen, right? For that, we use heptan. I
Hmm.
mean, an antigen which on its own cannot act as an immunogen. We add heptan and it becomes an immunogen.
Hmm.
So
So
for
basically
this question,
you have to understand that why adjuvant alone cannot be used. Tell me why, because that adjuvant molecule, because it is only increasing the immunogenicity, then why cannot we use just adjuvant? Why we need to have antigen attached to it at first place? Why can't we just use the adjuvant itself? Because adjuvant will not give us the specificity. The specificity would be provided only with the help of antigen, because antigen only knows where to bind. Antigen is actually the basic definition of antigen is that a molecule, a protein which binds to the antibody. That is just antigen. It can be immunogenic, it cannot be immunogenic. So adjuvant is, so you do not have specific and adjuvants for specific antibodies, no, but you have specific antigens for specific antibodies. So antigen will provide the specificity, and when it is combined with adjuvant, it will increase the immunogenicity.
ma'am,
Was
here the question is asking like regard to the generation of immunogen. But adjuvant, how can an adjuvant generate an immunogen? I mean, uh,
adjuvant will not generate an immunogen. See, adjuvant is not going to generate a different product. No, it is not going to generate a different product. Basically, immunogen means antigen, which antigen and immunogens are not different things. Okay, immunogen means that antigen antigen has the property of producing antibodies. Basically, what kind of immune response and we expect an antigen to produce? The kind of immune response that produce antibodies. That is the ultimate goal of providing antigens in vaccines, so that it produces antibodies. So basically, it is enhancing the ability, it is enhancing the activity of the antigen to create, even to elicit a response which can produce even more antibodies. So adjuvant is not creating a different product immunogen, no. Antigen adjuvant is basically enhancing the antigen in such a way that, and now this antigen which is connected to the adjuvant, this fused protein, is able to produce more antibodies. Now how you think how it can be done, how adjuvant can enhance the immunogenicity? Think.
ma'am, adjuvants can help the antigen stay longer in the body and let it not be degraded very
Yeah,
fast.
it, yeah, this point is there. So see, one thing is that first thing is so when you're giving your adjuvant, what you said is right. So that is the consequence. Basically, when you are attaching adjuvant to an antigen, first thing what you are doing, you are increasing the size of the antigen. Increase in the size will create more immunogenic response. You are increasing the complexity of the antigen. You are increasing the molecular weight of the antigen. So these three things, so by default it is happening only. And also the point that you mentioned, that there would be a sustained release of the antigen. So this is how adjuvant will kind of helps that particular antigen to produce an enhanced immune response. And when we talk about enhanced immune response, that means enhanced generation of antibodies. So basically, many kind of, even in many kinds of vaccines also, this the basically, what is vaccine? So vaccine is nothing but the antigen, which is in a form that it is able to generate as many antibodies that can generate an immune response in the body, but not that many antibodies that it will cause drastic negative effects on the body itself. So that's why when you take vaccine, you have a fever for some time, because you are actually getting that antigen. So if you are taking a vaccine for the virus, that means you are actually taking the virus in such a form that it is able to generate immune response in your body, but it is not harming your body also. Is it clear? That is what vaccine is. You took the vaccines, we all took the vaccines. So we all took the vaccines for COVID. So we actually took the virus only. So that is why, if you could relate, I don't know why it is advisable that when you already have fever, then it is advisable not to take the vaccines, even because of negligence. This happened in our country also, that many people, they people who do not have the basic knowledge of science and do not have the basic knowledge of how these things work, so they thought that vaccine is actually kind of medicine. So even in my known cases also, this happened, that when they got COVID, and then they thought that if we'll take the vaccine, then we'll be fine. So during COVID, they took the vaccines and they could not survive. Why? Because already the virus has attacked in their body, and on top of that, they took even more virus in the form of vaccine. So it created an enhanced response in their body. So that's why basic general knowledge of each and every domain of life, whether it is science, whether it is different fields, whether it is physics, chemistry, different different fields of science, or whether different any kind of basic knowledge is very important. So this happened in India with many people, that they confused vaccines with medicines, and they took vaccines when they already had COVID. So you should know that vaccines are just the virus, and that vaccines can be produced by many ways. You can heat inactivate it, or you can, there are many ways of producing the vaccines. But nevertheless, here what we are studying is that antigen, to enhance the antigenicity, or to enhance the generation of antibodies, antigen is mixed with adjuvant. Okay, the next question is, the equilibration reaction during affinity chromatography requires high salt. Why you think high salt is required? So the high salt is required because high salt decreases the non-specific interaction between the protein and the analyte. Okay, next question. We have to finish it because we just have half an hour and we are on the fifth lecture. We are not yet not at the fifth lecture. Anyways, so question number 14th. What are the conditions for storing the column? Column has to be stored in 4 degrees, in 20% alcohol with sodium azoide, stored at room temperature, A and C and A and B. This we have discussed many times, I think.
A and B.
A and B, yes. So it has to be stored at 4 degrees in 20% alcohol containing sodium acid. Then we come to the different types of affinity chromatography, that is bioaffinity chromatography and metal affinity chromatography. So bioaffinity chromatography, we have already discussed that here we talk about the enzyme and substrate kind of reactions, or antigen antibody kind of reactions. And metal affinity chromatography, there are nickel anti kind of interactions. There are many kind of interactions. We just quickly go through the different interactions. Okay, we get. So then there are many different kind of interactions uh, that takes place when we talk about bioaffinity chromatography. This is just like a general mechanism. You can go through and you can just have a look of what are the different kinds of different types of receptor and ligand interaction that takes place. So you have GST based purification, where glutathione, which uses the glutathione as a substrate for some reactions, so that can be used. Basically, you have to just remember that glutathione is used up by glutathione transferase, glutathione as transferase. So this transferases uses glutathione for the reactions. So basically, you can have GST and GSH, मतलब glutathione and glutathione transferase interactions, which can be used in affinity chromatography. Okay, so basically you have to tag your protein with GST. Okay, and then you can have the affinity flow, and then you can just elute the product. Then we come to metal affinity. Just a minute. Metal. uh, Okay. Mm. So I don't know why students are not coming for the classes. So every week I see the number is getting reduced only. Probably it's difficult to sustain the same enthusiasm for a longer period of time. During the initial classes, students were very enthusiastic to learn and to see what is happening. Now I think they are not that much enthusiastic, or I don't know whether you have any this thing connection with the other course mates. Do you know your other course mates who are in this course, who have enrolled in this course?
No, ma'am. Ma'am, but I think due to college and all, and also, ma'am, the videos are posted on Saturday or weekend. So, or by Tuesday, it's not possible for us to watch all the videos.
Then you think about me. I'm doing research. I
Yes,
have to
ma'am.
first I have to go through all the videos. I myself have to learn, matlab, not learn, but I have to revise everything whatever is there in the video. Then I have to make questions by myself. I don't know if I and I'm doing research. My research is not like a 9 to 5 or a 9 to 7 job. It's a full day job. Even if we have to go on Saturdays and Sundays, if we have some experiment running, then we have to go on Sundays also. So I don't know if I am managing, and I have, it's not just the only place where I'm teaching. I have to do some internal TA ships also and some other TA ships also. Anyways, so now we'll come to metal affinity chromatography. So in metal affinity chromatography, this nickel NTA chromatography is very popular, and it is important also. So many questions are often asked with this nickel NTA based column based chromatography. So it's more or less, it is just the basic principle is the same for each and every affinity chromatography. Just the name of the ligand, the name of the receptor, the name of the two interacting partners will change, and a little bit difference in how to create the columns and how to kind of couple them and charge them. That is a little bit different, but other than that, each and everything is quite similar only. And I think then we have the applicatory part of affinity chromatography. So so I think if there is any, if by chance you have any feedback form, uh, you can say that they can have a system where the videos are uploaded maybe on if they are uploading the videos on Sundays, then they should have a live class on maybe Friday, so that at least students have a week to uh, go through the chapters. um, or they can manage in some ways, because I think live classes happens on Fridays also, but um mine is Tuesday because I only chose Tuesday for my chapter. But I think then accordingly they should arrange the timing when they have to release the chapters, because in 2 or 3 days it's actually difficult to cover all the classes. And some weeks have more classes also. This week you had six this thing chapters. I don't know whether you have this feedback forms or not. Anyways, hmm. So then we have just brief different applications of affinity chromatography. So you can use affinity chromatography to isolate a particular protein complex based on the interaction that happens with the other molecule, with the other binding partner. You can have the isolation of protein complexes, then you can use some proteases assays, you can have, so there are different applications that are there. So first we'll solve the questions and then we'll try to conclude it here, and then we'll see what is there in the next. I think next you will also have some different techniques maybe. Okay, so now we'll do some questions again. Okay, so GST based purification utilizes GST tag on the protein, glutathione as a substrate for reactions, GHS presence on the bead, none of the above or all of the above.
All of the above.
Yes. The answer is all of the above. Can you explain how? Matlab, can you explain the importance of each and every point here? Just like basic. So GST is glutathione S transferase. So glutathione S transferase will bind with glutathione. So glutathione S transferase has to be tagged with the proteins that we have already learned. Now the this glutathione S transferase will bind to the glutathione. So obviously you need to have glutathione, and that glutathione is present on the beads, and that bead is basically the matrix. That means matrix will have your glutathione, matrix will have your GSH, and GST is present on the proteins, and the interaction between them takes place. So GSH which is present on the bead is basically matrix, and your GST which is tagged to the protein is the other one, and the interaction between them takes place. So it is a kind of bioaffinity chromatography. Glutathione as transferase and glutathione is a kind of bioaffinity chromatography. Then your antigen
Ma'am,
antibody.
won't there be any problem if our protein has this GST tag?
No, no, no. See, uh, uh, in experiments, when you do some experimentations in our field, we have a lot of tags. Okay, in not just in this purification part, but even when you are doing some kind of immunohistochemistry or immunoflow cytometry, even if you are doing, so there also you have to have various kinds of tags. You also have fluorescent tags. Even uh, just leave fluorescent, we also have radio label tags. But those radio label tags cause any problem to the proteins or matlab, to the molecules? No. So basically, so these tags are engineered, are made in such a way that they will not cause any harm, or they will not change the properties of the protein. So they are engineered and they are modified in such a way. So whatever tags that we use, we don't use them in their native form. They are very well processed and engineered so that they can be made in a way that can be used in experiments. So even you have fluorescent tags we have. So when we are doing immunofluorochemistry, immunofluorescence analysis, microscopy, if you're doing fluorescent microscopy, confocal microscopy if you're doing, so you have to have tags. Even when you're doing flow cytometry, even if you're doing fax, there also you have tags. So everywhere you have to tag them, otherwise how can you detect or how can you determine where it is present or whether it is present or not? So, the presence of or absence of any protein or any molecule is known only when it is attached to a tag. So, everywhere you have to attach a tag. Here also you have to attach a tag. But they are engineered and they are made in such a way that it will not cause any harm to the protein. Okay. So then next question is in nickel NTA chromatography. So this nickel NTA chromatography is a kind of
Met-
Bioaffinity chromatography, metal affinity, dye affinity. It's a
Metal affinity.
metal affinity chromatography. Okay. So now we are coming to the last part of affinity chromatography. So here we have covered the applications of affinity chromatography. So affinity chromatography can be used for the isolation of protein complexes. They can be used for protease assays. They can be used for immunopurifications, and there are many other more applications of affinity chromatography. So these are like the general applications or the known and very famous applications. But obviously the applications differ from the kind of experiments that you are using, the kind of objectives that you have. So it's very important to understand that when you are reading a particular technique, you read about that particular technique only. Like if you're reading about SDS page, then you will only read about SDS page. If you're reading about a gross gel, you will read only about a gross gel. But when you are doing it, you do it all together. You are doing a gross gel also, then you are expressing the protein, you are doing page also, you are doing one chromatography, you are doing another chromatography also. So reading and understanding the concepts is one thing, and implementing it on the bench is another thing. Therefore it's very important. See, you guys are taking this subject. It's a very good opportunity for you to get understand of the basic concepts, because if even if in masters, if you have this much of clarity, it's really good. But along with this, you also have to have some kind of experimental exposure, where you have some kind of real hands on training of how to perform, because knowing the concepts is one thing and doing it on the bench is another thing. So do you have like practicals in your classes, in your
Yes,
curriculum?
ma'am. Ma'am, SDS page we have done agarose, well, electrophoresis, and thin layer chromatography.
Okay, so these things you have done. Okay, so from which college you are?
Ma'am, Delhi University's Swami Shraddhanand College. I'm doing microbiology third year.
Okay, okay. So you're from D. Fine. So yeah, so you need to have some kind of exposure, and also like if you have summer vacations or winter vacations, you can always go for some internships. And I can even help you if you want, like how to apply to internships, how to find colleges, how to write to the different professors or scientists, if you really want to pursue higher studies, for if you want to go for research and all. Anyways, so now we'll come to the questions. So the first question is sodium dodecyl sulfate, which is used in SDS page. It's a cationic detergent, non-ionic detergent, anionic detergent, or it's an ion exchanger. Very simple question.
I'm a anionic detergent.
Yeah, so it's an anionic detergent. Then next question is anti-agarose beads are charged with
Nickel
NaCl.
sulfate.
Hmm?
Nickel sulfate.
Nickel sulfate. So the option is D. Hmm. So the next question is role of SDS in SDS page is to denature and impart negative charge, to impart overall positive charge, to impart equal mass to the proteins, or to unfold and impart net positive charge to the proteins.
Ma'am, option A.
Option A, denature and impart net negative charge of the protein. Yes. Now you have to, this is just a normal question. Calculate the KD value if elution volume is 10 ml, void volume is 2 ml, and pore volume is 1 ml. So first we have to see what is the formula. So what is the formula for KD? Here, first of all, tell me, here we are talking, where is the question? Yeah, elution volume, void volume, pore volume. So which chromatography technique we are talking about here? Which chromatographic technique we are talking about here in this question?
Ma'am, gel.
gel electroporation. So that means you have to use that formula. So because elution volume, void volume, these are the things which are in the gel electroporation. Sorry, gel this thing, chromatography. So quickly we'll see the formula. So formula is VE minus VO by VI. So it's easy. Calculate it. First calculate and tell me the answer. VE minus VO upon VI. So what is the answer?
8.
Yeah. So so this is like a very very basic question. So these kinds of questions, I don't think it will even come in examinations. It might come if you are giving DBT, then it might come in section A maybe. Maybe in section A it might come, but otherwise, so the questions would not be so easy. I just gave you one question to just to let you remind that basically from this whatever you have understood. So even if you have gone through your assignment questions, so you could see that the questions are very they're not very straightforward. I mean, they are conceptual and logical questions. So even little more difficulty level would be there, and obviously more difficulty level would be there in gate and net. So thing is that whenever you are learning these chromatographic techniques, you have to be very clear about the concept. Because if you know the concept, then whatever is the language of the question, how much the question is twisted, doesn't really matter, and you can go through them. But if you don't know the concept, then it will become very difficult for you to understand. For example, we are talking when we talked about gel filtration. So in normal filtration, so when you have a normal filtration, you have a filter. And I say, suppose if I say that this the pore size of this filter is 0.22 micron. So that means any particle which is above 0.22 micron, whether it will pass or it will not pass, it will not pass. It can only pass particles which are below 0.22 micron if I am talking about a normal filtration. Okay, so that means particles which are less than 0.22 or which are 0.22 micron, they can pass through the filter. But particles which are larger, they cannot pass and they will be here only. So here in normal filtration, elution, what is coming first in the elution? The smallest particle in normal filtration. So when you're talking about normal filtration, so smallest particles are eluted first. Okay, but here when we talk about size exclusion in gel filtration, smallest particles, smallest particles, they are eluted in the last. This is also a kind of, here also filtration is happening, but here the filtration is a kind of reverse filtration. So this is a very basic logic or a very basic concept that you all must know, that when you're talking about normal filtration, so when you filter anything, then the the molecules which are larger than the pore size, it will not pass. But when we're doing the size exclusion, the molecules which are larger than the pore size, they will only pass. Okay, could you understand, could you compare the difference between this two different filtration, normal and reverse? Is it clear?
Yes,
It is completely opposite. So this is one confusing thing. Another confusing thing is globular and fibrous proteins. You should understand that proteins can have globular conformation, it can have a fibrous conformation, but here mostly we're talking about the globular proteins because we're talking about the different radius of gyration, different radiuses. So radius means obviously they are globular proteins. Second thing is that whenever we are doing the applicatory part, so if the size is increasing, the graph will shift towards the left side. Always remember, whenever we are talking about gel filtration chromatography, if the size of any particular molecule, the size of the analyte is increasing, the graph will shift towards the left side. That means lesser time would be taken up, or lesser volume would be taken up for dilution. Okay. So if we are dealing about protein ligand interaction, there was one example of protein and ligand interaction. So if this is your one peak of protein, and this is your another peak of ligand, okay, and when there is an interaction between protein and ligand, then you have a peak somewhere here only, protein plus ligand. So with the help of peak, you can determine whether interaction is happening or not. Because you already have the information that protein will give a peak at this particular point, ligand will give a peak at this particular point. But if they are interacting, their peaks should be leftmost at the left side. That means they are, because when they are interacting, what is happening, their size is increasing, and because their size is increasing, the peak or the graph is shifting towards the left side. So these are very basic concepts that you have to remember about this thing, gel filtration and chromatography. So about the affinity chromatography, the basic concept is interactions. It is it is solely based on the interaction of the analyte with the matrix. If a mixture of analyte is passed to the matrix, then based on its affinity to bind to the matrix, the uh, it is the separation takes place. Now affinity chromatography, it can be bioaffinity, or it can be metal affinity or diaffinity chromatography. When we talked about bioaffinity, what we talked about enzyme substrate reaction. We talked that the reaction should enzyme should bind to the substrate, but there should not be any formation of product, because we want to isolate the enzyme or the substrate, whatever is there. We do not want to isolate the product, so there should not be any product formation. Then we have learned about antigen antibody interactions. So we have learned that how to produce the antibodies. So for the production of antibodies, you need to have first the antigen. Then this antigen is converted to immunogen. Then this immunogen is given to the organism, and then then you give the booster doses, and then you collect the blood and you take out the sera, and you isolate and purify the antibodies. So this is how your antibody production takes place. And then we also talked about in brief about the glutathione S transferase and glutathione interaction, which is also a kind of bioaffinity interaction. Then next kind of interaction is a metal affinity interaction. So in metal affinity interactions, basically very famously studied and very important is your nickel NTA interaction, that you can just have a look. And then in between we had some other basic concepts and topics related to how to do the receptor coupling, and what is a general flow of performing a particular technique, what are the advantages, what are the other applications. So this was all which was covered in the last class. So if you have any questions or doubts, you can just ask anything that you think is a little tricky, that you want something, you can ask me.
Ma'am, I had some general doubts, not with this topic.
You can ask. I'll see if I can
Ma'am,
answer.
you did bachelor's in which subject?
My bachelor's was in biotechnology,
Okay,
BTech
ma'am.
Biotech.
And then masters also did in biotech.
My master's was in animal biotechnology. Yeah.
Okay, ma'am.
Hmm.
Ma'am, so you gave gate and net both.
Yeah, so I gave GATE also, and I gave so basically I was more inclined towards the animal biotechnology side, rather than the plant biotechnology side. So I don't know if you know, do you have any idea about Indian Council of Agriculture Research, ICAR?
Yes, ma'am.
There is
Pusar,
one
ma'am.
PUSA is there in Delhi now.
Yes, ma'am.
ICR PUSA is there in Delhi. So
Yes.
I gave that examination also. So from there you have some ICR institutes. One is Pusa in Delhi, and another one is in IVRI in Izzat Nagar, Bareilly, and another
Yes.
one is in Karnal. You know Karnal? You are in.
Yes, ma'am.
So in Karnal there is NDRI is there, National Dairy Research Institute. So I did my masters from NDRI, Karnal. So then then I have worked as a JRF in Tata Cancer Hospital, that's in Mumbai. So there I have worked as a project JRF for two years, and then I joined here as PhD in University of Hyderabad. Yeah,
Okay.
that was just like my basic introduction. Anyway, so if you have any doubts regarding any any of the topics, or in general also, like if you want to apply, like what to do after your bachelor's, like how to opt a good university or a college, what are the things that you should uh, really see before opting for a stream, whether you have to go for plant or animal or microbiology, whatever it is, or maybe bio process also. So these questions, if you have any queries, you can mail me. I think my mail ID is now there with you all. I can try to resolve them, or you can just leave in the comment in the YouTube, or you can ask me here also, like whatever is feasible. So just like whatever I can help and guide you. That's our primary objective also. So like any kind of help if you want, you can always ask me and contact me. So
Okay,
I
ma'am.
think
Thank you, ma'am.
yeah, fine. So with this, I think we'll conclude today's lecture, and we'll meet again in the next class next Tuesday then. Okay. Okay. Someone has Naraini. Yeah, you can ask me, Naraini. Yes, you can ask.
Ma'am, right now we were talking about chromatography and page or like the gel electrophoresis.
Yes.
So recently, in our bioanalytics subject, we were taught about the same thing. So we did SGS page, but he didn't told us about the like a professor. Our whole experiment was wrong because we didn't know when to stop the phoresis. So our whole sample was like gone.
Was
So
eluted more. It just went off.
yeah,
Yeah.
like we didn't knew like we have to stop when we have to stop because it was the first time we did. So I want to ask you is now we know like we were given one plain sample of protein, but what if we have got different samples,
Hmm.
and like if we have seven sections in the electrophoresis and we have 20 samples, so like how you do it?
So I could not guess your question. So basically, see, whenever
I
you
was trying to say is that I have different types of protein samples, and each protein is like after we do the electrophoresis, the whole process is done. We have to stain them to see
hmm,
what how exactly it is going.
hmm, hmm.
So I want to ask like, is it a single dye for all the proteins or is it different dyes for different proteins?
Dyes, see, so the dye that you use, Komasi, that is same only, but how you have to determine that what is your what which protein is what, or what protein is what, is basically is basically depends on the size of the this thing, the protein. So whenever you are doing any electrophoresis, whether it's an gross electrophoresis or you are doing a page, so what is the most important thing? The most important thing is to have a comparison. So you have to have a ladder, you have to run some control samples. Okay. So many control samples, like depending upon whatever experiment that you are doing. So you have to have some control samples, you have to have a ladder with which you can compare ki acha, so this protein has given me, when I run this when I have run this, it has given me two bands of this and this size. So okay, from this I can determine that maybe it can be a heterodimer, or it can be, if a protein is giving two bands, so there might be a possibility it is a heterodimer or a homodimer depending upon the size of the protein. Or it might have also, it might also be a mixture where you're getting two proteins. So all this you are able to determine or to get only with the help of a control that you are running. So whenever you are performing any experiment in biotech, in life science, or in general any experiment, you always have to have a control with which you can compare. Okay, so yes, Karthik, you have raised your hand, you can ask.
Ma'am, I just wanted to ask Narayani, did you use bromophenol blue as the tracking dye?
No, no, we use chromosine blue.
So
Like
Kumasi
that.
blue is used to visualize the protein, but
Kumasi is
didn't
used
you
for
add?
visual. It is at the end. So Kumasi is like for the visualization in the end to see whether. So have you done this thing? I don't know if you know. Have you done Pochu staining or something like this?
What, ma'am?
No,
Pochu.
ma'am, no.
Have you done that? Okay. Okay. So what we can do is, I think there are, before doing the experiment, you should also have like a basic. You should have to have the basics of why we are using what we are using why.
No, no, we had the lecture. Like we usually our syllabus and the practical. We have it side by side going. So like that experiment was fail. So we didn't got to know what exactly our result was. So like we did it one more time like the other batches. But so we didn't did it. So if
Fine.
like
See, I don't know how you perform the plotting experiment, but in general, if sir has given you, then obviously it must be an optimized, standardized procedure. But in general, whenever you're performing any experiment, suppose you have a protocol manual. Here they have written you have to use it this much percentage of this much percentage of stacking, this much percentage of resolving, whatever it is given. But you always have to optimize, you always have to standardize the percentages and everything according to your particular proteins. So it's not like ki whatever is given in the protocol that will work always. No, it never works in
but
reality.
what if the sample is unknown? Like we don't know that if it's glucose or sucrose or maltose.
Yeah. So see, so when you are doing an experiment, like I don't, I really don't know how you kind of started and what was your initial sample or whatever. But like if you are doing, suppose you are starting, so you will have a little bit of idea of what kind of proteins you want to isolate. So at least what kind of proteins you want to run. Or if you even don't, even if you don't have the idea, so at least you will have an idea ki these proteins I am isolating from this substance, so it can be this, it might be this. Okay, first thing. Second thing is that SDS is not going to give you all the answers. SDS is not going to solve all the queries. It will not going to tell you the exact complete information of the protein, it is this, it is that. It will only tell you, depending on the molecular weight, what is the molecular weight of the protein. And you can determine what how many subunits are there. You can determine whether the oligomeric status also you can determine, if you know like how many subunits are there and whatever. So that much query only will be solved with SDS. What are the different properties of the proteins, with what different kind of protein is there. For that you need to have a series of experiments subsequently, where you can try to determine the more, even more characteristics of proteins. Okay, so any particular experiment has its own limitations, that it can answer your query up only up to a limit. It cannot, just one experiment will not give you each and every property and each and every information about the proteins, right? So SDS will only provide you the information about the size. You already knew which molecule you have, I mean which molecule you are using. So the size also you know. But I mean, so I don't know. So you know actin.
Yeah.
Yes,
So basically when we are doing SDS, so we kind of use actin as a control also. So you have to have some controls. Okay, so with the help of those controls you can kind of compare your bands, whether they are coming or not. Also, so when you're doing SDS, it's not just doing the SDS, it's also to have a proper band. So what I mean by proper band is that your band should be free of contamination. So your lane should be very clean. So whenever you are STS, your lane should not have secondary bands or some, I mean, it should be a very clear band when you're doing STS. So again, that needs a lot more of standardization processes and everything. Yeah, Karthik, you can ask.
Ma'am, I'm sorry for dragging the meeting, but I think I understand what she's trying to say. So,
Okay.
I mean, she's asking that we have a variety of protein samples. I mean, some protein could be really small and some could be really large.
Yeah.
So, if we wait for the large protein to move ahead, so the small one could just run off the gel.
Yeah.
So,
Yeah. So see, that is what I'm saying. Yeah. So, okay, okay.
and
Your
I think I also want to ask them why didn't they use the tracking die? Because the tracking die will always be ahead, and so you can actually figure if
yeah.
the protein is going to like.
What we did was we used the tracking die and all, but after the sample ran off, the sir came and he was like, "You have to stop it." So we were like, we stopped it. We removed the gel, and we dyed it. But the result, what was the exact result, we couldn't see because we had our lectures, so everyone had to go down.
So
So
first
your
thing,
tracking that die mark, where was it on the gel?
it was first time we were doing ना, so certain said that यहाँ पे रुकना है. So हम लोग कंफ्यूज हो गए कि हमें अभी स्टॉप करना है या सर आने के बाद स्टॉप करें. तो वो निकल गया पूरा सैंपल.
Hmm.
see, you have to understand that when you are doing SDS, see, that's what I was telling you now. Ki, see, it's you have to design your experiment in a way ki you're not, ki if you can have a possibility that there are so many proteins, some are large, some are small, there is a great variation, then you should not do SDS at first. You should do some other filter technique, any some other technique that can filter some molecules. So in general, when you are doing SDS, you don't really have like a mixture of proteins, and you don't really have like numerous bands which are coming in just one lane. You don't do SDS for such means. So you basically, in normal, you use SDS when you really want to have like the size of one or maybe two maybe, but not like a mixture of proteins you're putting. So in, I mean, in practical, I'm telling you, okay. So again, as I said, key theory and practical, they are very two different, entirely different things. If you have an experiment planned out, if you have an experiment and you know that this kind of problem can come in your experiment, if you are speculating it, then why not to exclude some of the this thing molecules which are not very important for you. Someone
Ma'am,
has joined.
also they can use that gel where there is this gradient. I mean,
Yeah, they
from
can,
in the
they
beginning.
can, they can use, you can even use the gradient also. Yeah, that's also a very good point. You can even use the gradient. So anyways, it depends like the kind of experiment that you have planned, the kind of molecules that you want to isolate. And when you are doing a protein purification takes hell lot of time. Okay, it's not like a one day job or one experiment job. There are so many factors that has to be controlled. you, y- so many kinds of techniques, so many kinds of parameters and filtrations. At many times they also denatures the proteins also, because you have to not just isolate the protein, you have to isolate the protein in its original conformation also. You cannot denature it by any means. So that adds up to another level of problem in it. So anyways, this discussion is, I think, it's an ever going discussion. But it's good that you have queries and you are coming up with the question, the queries, whatever you have faced in your practicals and theory. I appreciate it. So I really like this kind of interactive class. If you also come up with questions, and we can have a discussion, as we all are learning from each other. I'm not just a teacher, I'm also a student. Maybe I'm a PhD, you are you are bachelors, but still we all are students, we all are learning. So if we have good healthy discussions where we are discussing science, we are discussing the problems, it's always welcomed from my end. Okay. So okay, then we'll stop the class here. And if
Thank
you
you,
have any
ma'am.
yeah.
Thank you,
Thank you,
ma'am.
ma'am. It was really a useful session for us also.
Okay, then it's very good to hear. Okay, then, so bye bye, we'll meet in the next week.
Bye, ma'am.
Yeah.
Bye, ma'am.